Hello everyone. Today we are going to see scanning electron microscope or SEM. SEM is a spatial microscope that uses electrons instead of light to give us a very detailed look at the surfaces of different materials. Imagine looking at tiny particles like grains of sand or cells with such detail that you can see every bump, groove and shape. That's what SEM helps us do. And it's used in many areas from studying biology to developing new materials. To guide us, here is an outline of what we will cover today. We will start with where SEM is used, look at its main parts, and talk about how we prepare samples for it. Then we will learn about how SEM works, its pros and cons, and where it is commonly applied. By the end, you will have an idea of how SEM helps scientists and researchers. Okay, so what is SEM? SEM is a short for scanning electron microscope. Unlike regular microscopes that uses light, SEM uses a beam of electrons to scan a sample's surface. This gives us super high resolution images, meaning we can see tiny surface details that we wouldn't be able to see otherwise, whether it's a metal, a plant, a tiny insect, or whatever. SEM can show the 3D images, surface texture, and even some of the elements present on the surface. Now, where is SEM used? It is useful in field that needs to see really fine details. Some common uses include shape and topography. It shows the 3D shape of the surfaces. Texture, we can see how rough or smooth something is. Composition, it helps identify the elements present in the sample. Crystallography, for materials like metals, it can show the arrangement of atoms. This makes SEM important in material science, biology, chemistry, and electronics as well. SEM has two major parts, electric console and electric column. Electric console control the knobs and switches that allow for instrument adjustment such as current, magnification, brightness, contrast, etc. For electronic column, it is generated under vacuum conditions, focused to a smaller diameter and scanned across the surface of a specimen. Now let's talk about the main parts of an SEM. Electron gun. This is where the electrons come from. The gun fires a beam of electrons that will scan the sample. Lenses. This works like a camera lens to focus electron beam down to a very tiny point. Scanning coils. These move the electron beam across the sample surface like a scanner moving line by line. Sample chamber. This is where we place the sample and it keeps everything still for a clear picture. Detectors. This pick up the signals from the sample and then turn them into an image. Vacuum system. Because electrons can't move well in the air, we need a vacuum around the electron beam so that it stays focused. Uh, like I have already said, electron gun is used for producing an intense beam of electron. For this, thermionic gun is generally used. Majorly, two types of electron guns are used industrially. First is the tungsten hairpin electron gun, and the second one is lanthanum hexaboride electron gun. Anode plate is a metal plate having a positive charge. It attracts electrons to form a beam. Majorly, two different lenses are present in an equipment. First one is the condenser lens, which reduces the diameter of the electron beam, and the other one is an objective lens, which focuses electron beam. Scanning coils are used to raster beam across the sample surface so that whole electron beam can scan the entire sample. They are able to move the beam. Sample chamber is where the sample is placed and can manipulate and can move the sample. Detectors pick up the signals from the sample and then they turn them into an image. Because electrons can't move well in the air, we need vacuum around the electron beam so that it stays focused. That's why we need vacuum chamber in an equipment. For dry sample, first vapor fixation is done, then it is mounted, then coated, and then finally SEM observation is done. For the wet sample, first chemical fixation is done. Since the sample is wet, so it has to be dehydrated. So first dehydration is performed, then drying is performed, then the sample is mounted, coated, and then finally SEM observation is performed. These are some of the conventional SEM specimen preparation for the living tissues. You can see from this slide. Following are some of the important parameters in preparing a sample for SEM imaging. We need a very clear sample which is free from dirt and dust so that we get a very clear image. For biological samples, we need to use appropriate buffers or distilled water. Surfactant can also be needed in some of the sampling preparation techniques. Use of ultrasonic system can also be given importance in preparing sample. However, ultrasonic baths require caution in order to avoid damaging the sample. For biological samples, we use chemicals to preserve their shape and remove water so it won't interfere. For dehydration, use a graded series of alcohol and finish off the final dehydration step with 100% alcohol or acetone. The sample must be completely dry to avoid vapor that can mess up with the electron beam. So, before placing the sample in high vacuum environment, it should be dry, as I have already mentioned. Otherwise, water vaporization will obstruct the electron beam and can interfere with the image clarity. While using biological samples, we should be careful when doing CPD, that is critical point drying, so as to not compromise the structural integrity of the sample. For non-metallic samples, we add a thin metal layer like gold to help the electrons to create a clearer image. It also makes non-metallic samples conductive. Here, we uses argon and electric field to tear off metal from cathode. Also, the metal falls onto the sample and coat the material. So this is how sample is prepared. So, uh, summarizing the sample preparation technique. First, for living tissues, fixation of fluid is done. Then, further it is rehydrated. Then it is dried. Then it is mounted. Then the sample is coated. And then finally, SEM analysis is done. Here is how SEM actually works. The electron gun fires a beam of electrons at the sample. This beam is focused down to a tiny point by lenses and it scans the sample surface line by line. When the electron hit the sample, different types of signals bounce back like secondary electrons, backscattered electrons, X-rays. The SEM detectors collect the signals and then turn them into a digital image which we can see on a computer screen. When the beam of electron, that is an incident beam, incident on the or fall on the sample surface, different kinds of electrons get emerged from the sample surface like backscattered electron, the electrons which are deflected back in the direction of beam, secondary electrons when the ionization of electrons takes place in the specimen atom and these ionized electrons leaves the atom with a very small kinetic energy. X-rays, bombarding electrons displace an electron from the shell and at the same time another electron from outer shell occupies that space. During the replacement of the displaced electron, photons are liberated. Auger electrons, these are the special types of low energy electrons that carry information about chemical nature, atomic composition of the specimen. Cathodoluminescent, this effect results when the energy of impinging electron is converted into invisible light. We have already covered the working principle of scanning electron microscope. You can see from this image also. SEM is amazing, but it has some limits. We can only use SEM on solid samples, not liquids or not for gases. The sample size has to fit into this SEM chamber. It can't detect very light elements like hydrogen or helium. The sample has to be in vacuum, which can be tricky for some materials. This is a column and this is a specimen chamber and this is a cover plate. If we look at the longitudinal section of this instrument, then at the bottom side there is a high vacuum pump. This high vacuum pump creates a vacuum in the column and the specimen chamber. Creating vacuum is necessary because it removes any air particle or dust particle that is present in an environment, which will hinder in the quality of image which the computer will produce. Then there is a specimen stage where we can place our object of interest or sample of interest. This specimen stage can move left to right, upward or downward. At the upper side of the column, there is a V-shaped wire which is placed, which is mostly made up of tungsten. You can see in this video. This V-shaped wire is called as thermionic cathode. When high electric current is provided to this thermionic cathode, then this cathode generates a series of electrons. After this thermionic cathode, another plate called anode is also placed in an equipment. This anode is connected to the positive pole and the thermionic cathode is connected to the negative pole. Because of the strong electric field which is generated because of this, these generates or accelerates the electrons that are generated from thermionic cathode. All these generated electrons are called as primary electrons. These primary electrons falls on the specimen. The next part of this instrument is called as electromagnetic lens. This electromagnetic lenses focus the beam of electrons on the specimen so that it directly falls on the specimen only. When this electrons gets heated on the specimen, then as the specimen is made up of molecules and molecules are made up of atoms, when these beam of electrons falls on the atoms, the electrons present in the outer shell of the atom gets knocked out by this beam. This blue color electrons that are shown in this video are called as secondary electrons. Another important part of this instrument is an electromagnetic deflector or raster scan detector. You can see in this video also. So this is a raster scan generator. This is also called as SE detector, that is secondary electron detector. This secondary electron detector is connected to our computer. It sends signal to our computer of how many electrons that had been detected. Based on this, an image is generated on the computer screen.